Good morning. Good morning, everyone. At least if you're joining us from the East Coast of the United States. To the rest of you, I know it's a different time of the day. Welcome all of you to the 6th Patient-Driven Precision Medicine Conference, our 6th annual such conference. My name is Isaac Kohane. I'm the Department Chair for the Department of Biomedical Informatics here at Harvard Medical School. And I have the pleasure to host this meeting as I've done in prior years. I'm very sorry to have to do this virtually for the first time because of the pandemic. But as you'll see throughout our panels, and especially in the final panel, this conference really is informed by the pandemic, both in terms of sociology and in terms of science. I would also like to thank our two very generous sponsors, Merck and Medidata. They have been steadfast in supporting our educational activities to advance science, and they would have been happy to pay for your breakfast and lunch, but unfortunately, they've just been able to help us support the media services and the organization of this conference, for which we're very grateful. So it's my pleasure to introduce, before our keynote, the Dean of Harvard Medical School, an eminent scientist, a member of the National Academies. He, Howard Hughes investigator, a former chief resident at Massachusetts General Hospital, a leader in stem cell researcher, a major supporter of data science and our Department of Biomedical Informatics, and who has been a professor of biological chemistry and molecular pharmacology at Harvard Medical School since 2010. And he's graced us with his opening remarks in prior years, and I'm glad to say he's able to do so. So please welcome George Daley. I'm sure he will hear mentally the appropriate applause. Thank you.
Good morning, everyone. I'm thrilled to be part of this wonderful event, even if not exactly thrilled to have to participate virtually. A sign of the times indeed. COVID-19, the once-in-a-century public health crisis engulfing the globe, has created a whole new set of epidemiologic, scientific, and clinical challenges for us to solve. It's opened up new research frontiers for us to tackle, and at the same time, it's forcing us to rethink so much more. This grim contagion is raising existential questions about models of healthcare delivery, about the role that social and economic structures play in amplifying health inequalities, and the resulting health disparities that magnify disease risk and influence disease outcomes in certain populations. This pandemic is forcing us to face our global interconnectedness, to re-examine just-in-time business practices, and to contend with our collective dependence on global supply chains for chemicals, medicines, and equipment. The crisis is reshaping the way we think, and if we are smart, the way we will act upon the painful but valuable lessons that COVID-19 is teaching us. But while SARS-CoV-2 has unapologetically dominated our attention and occupied our minds in the last 3 months, all the other scientific challenges from the pre-COVID-19 era are still very much with us. These are the challenges that drove us to pursue careers in science and medicine, in much the way that COVID-19 is now inspiring the next generation of researchers and clinicians to put their shoulder to the wheel and heed the call of duty. These are the challenges that continue to motivate us every day. And one of these challenges has unified all of you on a quest and brought you here today, the future of hyper-individualized medicine. Now, I see several points of intersection between the challenges posed by hyper-individualized medicine and those raised by SARS-CoV-2. Each patient with an undiagnosed rare disease presents to the scientist and clinician a biological mystery in much the same way a novel pathogen does. For what is SARS-CoV-2 if not a biological novelty on a global scale? The precise tools and techniques needed to solve one may differ from those needed to solve the other. Yet I do believe that fundamentally, the solutions on both fronts will demand from us a tried and true formula. The building of bridges across labs, researchers, institutions, and disciplines, the meshing of various modes of scientific and indeed various modes of intellectual inquiry, the cross-pollination of efforts across various players, academia, industry, regulatory, government, citizen scientists, patient advocates, and foundations. Whether we're dealing with an emerging pathogen or trying to design a treatment for an ultra-rare disease, we cannot go it alone. Individualized care has become the crowning glory of medicine ever since William Osler cautioned his acolytes that the good physician treats the disease while the great physician treats the patient with the disease. Could Osler have imagined that a 6-year-old girl with a rapidly progressive neurodegenerative disease could one day be treated with a custom-made therapy designed to correct her unique genetic error? What was the stuff of science fiction only a few years ago is now a reality made possible by the power of collaboration between academic researchers and federal regulators, propelled by patient advocacy. This was the fruit of the melding of different modes of inquiry with neurogeneticists and pediatric neurologists at Boston Children's Hospital, using insights from genomics, biochemistry, and machine learning to identify the precise genetic glitch underlying a child's disease and to design a treatment. This form of hyper-individualized care is Osler redefined for the 21st century, a magnificent story of collaboration and a story of therapeutic translation, molecular diagnosis informing rational drug design, drug testing, and the manufacturing of a medication made for a single patient. But while this achievement makes us justifiably proud, it also humbles us into asking some inevitable questions. How do we replicate this? How do we scale this made-for-one approach to make it feasible and practical for the millions of people living with orphan diseases? How do we overcome the regulatory burdens, manufacturing limitations, and cost barriers associated with the development and delivery of such treatments? Even more ambitiously, this case forces us to ponder, might there be a path toward adapting this approach for the treatment of more common diseases? Certainly, the industrialization of genomic technology and big data have laid the foundation for doing so, but how to actually make that happen remains as much of a mystery as ever. And in that respect, I think the quest towards mainstreaming hyper-individualized medicine raises some of the same fundamental questions posed by our efforts to vanquish COVID-19 and to create rapid response systems that prepare us for emerging pathogens in the future. Now, in both scenarios, we have critical pieces of the puzzle, but we are missing others. That is why I think our experience with COVID-19 represents a tale of hope and at the same time, a cautionary tale. On the one hand, we have tools and knowledge gleaned from work with previous coronaviruses, particularly around the structural configurations of the spike protein and its interaction with the ACE2 receptor, its entry point for invasion. We have the expertise and systems in place to define the basic biology of the disease and to unravel its pathophysiology. We have the capacity to design treatments. We have unprecedented capability to image, study, and model protein structures and protein interactions. We have the capacity to sift through existing drugs and compounds for antiviral properties. We have massive libraries of proteins and antibodies. We can do in silico screening of compounds to design small molecule treatments for viral disease or precision targeted therapies tailor-made for patients' individual genetic errors. On the other hand, in both hyper-individualized medicine and pathogen preparedness, we face some acute systemic problems. With COVID-19, we know much less than we should have and could have known about the pathogen's behavior and the pathophysiology of the disease. for at least two reasons. First, as a global community, we largely abandoned biological surveillance efforts and rigorous basic virology study of coronaviruses after SARS and MERS. Now, this departure was not at all driven by a lack of desire to pursue such knowledge, but rather by rapidly shifting funding priorities and nearsighted vision. Second, once the pathogen was afoot, as a global community, we did not take full advantage of clinical electronic records, and as a result had to play catch up clinically, yet another systemic deficiency. Now, clinical experiences from the bedside, when analyzed in aggregate, can provide invaluable real-time data about disease behavior. At their best, EMRs are living clinical repositories of data that give us rapid, iterative knowledge that is constantly updated to inform clinical decisions on the go. Think of the first reports of blood clots in relatively young and healthy patients with COVID-19. Think of the kidney failure cases that were cropping up among people with what had been deemed mainly a respiratory disease. These seemingly isolated cases could have been spotted for the telltale trends they were much earlier, if only we had in place the well-harmonized EMR systems we'd envisioned years ago. Now, the reckoning of this critical deficiency has sparked new efforts. A COVID-19 data collection project led by Zak Kohane and his colleagues is underway and has amassed electronic records from thousands of patients from nearly 100 hospitals in 5 countries. And patterns have started to emerge, such as shifts in D-dimer values, harbingers of coagulopathy, or changes in creatinine levels that herald kidney dysfunction. Striking examples that show sharing data across proprietary health record systems is not only possible, but can illuminate trends to improve public health outcomes. I cannot help but wonder whether this sort of hindsight might hold a lesson for hyper-individualized medicine on the value of data systemization and more. We must have the humility to recognize our weaknesses at the same time we recognize our strengths. Yes, we have the knowledge, expertise, and capacity to define the biology of disease and design treatments, but it seems to me that we have serious pain points in the systems designed to translate, operationalize, and deliver the benefits of this knowledge. Whether we are dealing with a new pathogen or an undiagnosed rare illness, we must act on the two fronts in parallel. We must solve the acute challenges in front of us, but we must not lose sight of the long game in our search for one-off solutions. Both scenarios present a problem. They pose a question about scaling up the benefits of these efforts, whether it's mass production of antivirals or vaccines, or the ability to quickly manufacture a precision-targeted therapy for a single individual and establish a platform that can be used for others. Of course, both COVID-19 and hyper-individualized medicine engender serious biomedical dilemmas. Who do you treat and when? Who gets a ventilator, the first batch of precise vaccines, or a custom-made therapy? At their core, these dilemmas all require hard decisions about resource allocation in times of scarcity. I do believe that the ultimate frontier of translation in both COVID-19 and hyper-individualized medicine will be the democratization of access. Both hyper-individualized medicine and COVID-19 therapeutics raise what could be the most critical question. How do we make these treatments affordable and available for everyone? How do we take the wonderful discoveries that we are making and will make in the lab and the clinic, and how do we propagate them globally as treatments available to everyone, not just for some? This should be the true benchmark of our success as physicians, as scientists, and as leaders of industry and of society. Both COVID-19 and hyper-individualized medicine present us with threats that within themselves also contain opportunities and chances to improve. It is not coincidence that the two characters that make up the Japanese word for crisis individually mean danger and opportunity. Our very mission as an institution was built to deal precisely with such challenges, whether they come in the form of a novel pathogen or an unknown genetic disease. And our collective charge has also never been clearer: generate new knowledge and harness it into therapies that transform human lives for the betterment of all. Thank you very much.
Thank you very much, Dean Daley. No, thank you for stealing all my comments, but I appreciate the vision. Actually, I do want to interject before I dilate on those comments to say that we all have been seared, as I'm sure you have, by the recent horrible events that we can't ignore, that present threats to liberty, opportunity, and equality, and that have their roots in racism. My own parents in Europe witnessed firsthand the fatal consequences of silence and passivity in the face of such bigotry. Dean Daley, in a separate missive to all of us, has asked for, across the school, 8 minutes and 46 seconds of silence to observe our solidarity with the family, with those who are threatened by this, in solidarity with the shutdown STEM movement. And we'll do that at noon. That's 12 p.m. Eastern Standard Time. And I want to thank you for Dean Daley's leadership in this regard. So, one of our, in fact, our first keynote speaker for the first annual Precision Medicine Symposium that occurred 6 years ago, keynote speaker was Matt Might, himself at the time a professor of computer science. Since then, he is now the director of the Precision Medicine Institute at University of Alabama. And during the keynote, he said, and I'll check my notes and make sure I get it right, data is the most powerful therapy of the 21st century. And those prescient words are all the more evident now, as Dean Daley himself just now said. If we had only known earlier what was actually recorded and known, for example, in the electronic health record system, we could have acted with much more alacrity in responding to a clinical course that we didn't know, but could, by the events happening in hospitals worldwide. The flip side is that when we do have the right data, when we do have the right instrumentation, things accelerate. We can see now the truly unprecedented speed with which we are developing vaccines, based on not only the urgency, but the fact that we have these databases, the crystal structures of the viruses and the proteins that they dock with. And we have worldwide sharing of these data. And we're able to engineer directly to protein targets for our vaccines. And that's what happens right when we actually align our data with our healthcare and our science, as we always should. So the conference goal that we have every year is actually to accelerate this change through bringing data together to treat patients appropriately. And that's really hard. It means doing something very, very difficult, which is to figure out precisely the largest group for which there is an effective therapy for a disease that ails you. But that's sometimes very difficult, because as we enter the genomic era, sometimes the largest group is one person who has a mutation that can be actually treated with a unique therapy, with a string of DNA, an oligonucleotide that uniquely treats their disease. And there can be no better example, perhaps, of this hyperindividualized therapy than the journey of Mila, who has Bann disease. Mila, who had this journey with her doctor, who you'll meet, Timu, but perhaps most importantly with her mother, Julia, who is our featured keynote speaker. The three of them created an amazing team, and I don't want to steal the thunder from Julia, but did things that universities plus governments are hard pressed to do, and they did it within a year.